So in this section we look at the large biological molecules, or the macromolecules. So up until this point what we've looked at in the chemistry is just really small things. Now we're looking at big things that you find in living cells. So we're getting bigger, we're getting closer to the cell, which should be the next chapter in there. So the four main classes of these molecules are going to be the carbohydrates, or your sugars, lipids or fats, proteins, and nucleic acids. Nucleic acids are what you have in your genetic material in there. And so their architecture or shape explains how they work in the body. So a lot of times with the smaller molecules you have actual chemical interactions that go on, but when things get bigger, a lot of your interactions actually occur more like a lock and key mechanism, where it's based on their shape and going in and turning things. So it isn't that you don't have the chemical reactions, but you also have things that are a little more mechanical in nature like that are based on the shape in there. So they'll form polymers, which are going to be long molecules consisting of many similar or identical building blocks. So it's kind of like Legos, where you stack lots of pieces together. That's an example of a polymer being built in there, and that's the shape of a lot of these things in there. The monomers are going to be the repeating units of the smaller molecules that serve as the building blocks, and so some of those will have functions of their own. This here shows where you've got a short polymer here in there and a longer polymer in there, but you've got the repeating building blocks in here. So one way that these are made is through dehydration synthesis. So dehydration, when you dehydrate something, you pull the water off. That's what you're going to do in a dehydration synthesis, is in putting it together, these two pieces together, you're going to pull off two hydrogens and an oxygen in there and get water out of the deal. And when you pull off that water, you end up with them all being bound in there. So if you go to split down a polymer, a lot of times we'll do hydrolysis in there. So the last part of that, lysis, means to split something, and that just means we're going to split it with water. So we put a water in and use that to split the bonds in there. So they can be formed with dehydration, split with hydrolysis in there. So sometimes you'll see it being referred to as a condensation reaction in there, where two molecules will be covalently bonded through the loss of water. So it condenses them down in there and you'll lose the water. So that's what happens in the dehydration reaction. So you may see it referred to either way in there. A lot of times with the macromolecules, they're going to be really dependent on enzymes to have their reactions take place in there. An enzyme is going to speed up a chemical reaction in the cell. So the carbohydrates are going to include your sugars and polymers of sugars. So the term saccharide also means sugar in there, and these are going to be your monosaccharides, disaccharides, polysaccharides. Mono is one, di is two, poly is many in there. Some of them are going to be aldehyde sugars and some will be ketone sugars. So an aldehyde sugar has this carbon double bonded to an oxygen on the end. A ketone sugar has it in the middle, is the difference in there. And when it's not really the kind of thing that you're going to have to worry about, like trying to look at them and identify the difference of them. So here's an example: glyceraldehyde and dihydroxyacetone. Any idea where you might find dihydroxyacetone? In what? Now, this is actually the sugar that's in self-tanner. So, and it has its own kind of yucky smell. That's why most self-tanners have a really distinct smell to them is this stuff in there. So a pentose is going to be a 5-carbon sugar in there. So penta meaning 5. And sugars a lot of times are going to end in OSE in there. Up here these were trioses. So here you've got ribose and ribulose in there. One's the aldehyde, one's the ketone sugar. The hexoses, you've got glucose, galactose, and fructose in there. Again, you've got your aldehyde and ketone sugars in there. I'm not going to expect you to remember the difference between an aldehyde and a ketone sugar in there. I can't think of one instance where I've really needed to nail the difference in there. So what's going to make up the difference in your sugars is basically the number of carbons that are in there. Also, is it an aldehyde or a ketose in there? But its name is largely based on the number of carbons and the spatial arrangement of it. So monosaccharides are a major nutrient that is used for energy of cells. It's also used for a building block of other macromolecules. So this is how you get the majority of your energy in your body, is through carbohydrates. It is the preferred source of energy. If you want your body to run well and things to all function well, you give it carbohydrates in there. That's what the brain needs to run on in there. So if you're hungry, what's it like to try and make decisions when you're hungry, or to try and concentrate? Yeah. And a lot of times you'll sit there and you go, well, I just can't concentrate or remember it in there. And it's because your brain wants to run on glucose. If it has to start breaking down other things to try and feed the brain, it's not real happy with that. It will run, it will keep your heart going and your lungs going, but the higher functions of making good decisions with the brain are not going to happen as well as they do in there. So what does that kind of imply to you that you should do before you take a test? Eat breakfast. I mean, even if you don't like to eat in the morning, at least have some juice or something to give you some carbohydrate to fuel your brain in there. Yeah. You actually need a mix of them in there. It depends on the situation. If you need energy now, that's when your body's going to want those. The problem is they taste good, really good. So we will tend to choose those over other things. And it's not that they're bad, it's just us liking them and not having the self-control in there. So if you look, like a bowl of legumes like beans in there. You know, beans can be good too. Or a bowl of, I don't know, say lettuce or cucumbers or something. Or a bowl of M&Ms. If you're really hungry, which one's going to look more appealing to you? The M&Ms. So we'll say you're on a little diet plan and you're thinking, well, you know what, I'm going to have the willpower and I'm not going to have any of those M&Ms. You eat the whole bowl of cucumbers and lettuce and everything in there. And what happens right after you finish that? You're still hungry because it takes a while for those to break down. So that's kind of where, if you're really hungry, if you have a few of those M&Ms and then left behind, what you'll do is you'll break up down the M&Ms, and as the M&Ms start to wear off, that's when you can start to get the lettuce out of those other, or the energy out of those other things like the lettuce and cucumbers that took longer to break down. So there's a different purpose in there, but the problem is if you aren't looking at it and thinking, I don't want to gain weight from this, that bowl of M&Ms looks really good. And if you're like me, if somebody does not take it away, I'm going to have just a couple more and just a couple more until they're all gone. It's actually embarrassing if I go to somebody's house and they have a candy jar. I have to take it and put it away. And I'm like, trust me, you don't want to leave this out with me around because I will finish it off. So your disaccharides in there are going to be 2 monosaccharides joined together in here. So we've got 3 of those that we see in biology. Maltose, which is 2 glucoses hooked together. Lactose, which is a glucose and galactose hooked together. And then sucrose, which is fructose and glucose hooked together. Any idea which one of those is table sugar? Sucrose is the one that's table sugar in there. So fructose is in most fruits, except grapes have glucose in them. They're a little bit different in there. And so also the effects on blood sugar, how quickly it raises the blood sugar. Fructose tends to be lower. It does not raise the blood sugar and fluctuate as much as sucrose does, except when it's in high fructose corn syrup. There it's been modified and it's not the same. Then it tends to actually have worse effects on the blood sugar. So most of where you get your fructose from, unless you're eating fruits, anything that's packaged is going to use high fructose corn syrup in it because it's much cheaper to sweeten things that way than it is with sucrose. So your polysaccharides, multiple monosaccharides joined together by glycoside linkages. So these are used for storing or building materials. So starches are going to be stored glucose polymers in plants within the plastids. And so this also includes the chloroplast. So when you eat plants, you're going to be getting some of the starch from it in there. Glycogen is the polymer of glucose that's stored in animals. So that's what we're going to store in muscle and liver cells in there. So when you go to do an activity, you're going to be using a lot of the glycogen that's in your muscles to fuel that exercise in there. And eventually you use it up and then you have to start relying on other things for sources of energy in there. So how we link these starches together is going to be part of what is going to cause the variation in the different types of starches that you have out there. So most of them are going to be joined by what we call a 1,4 linkage. So that's carbon 1 attached to carbon number 4. So with maltose, it's going to do that, and the angle of them is going to make them helical. How bone got in there, I don't know. So the angle of the bond is what makes them helical in there. So amylose, it's a simple starch in there. It's unbranched in there. Amylopectin has branches with 1/6 linkages in there. So when they're branched, they tend to be a little bit more complex to break down and going to take a little bit more energy to break them down in there. Structural polysaccharides in there. Cellulose is a major component of the tough cell walls in plants. It's going to have the 2 rings of glucose form alpha and beta rings in there. The starch is all alpha, but cellulose is all beta in there. And because of it being beta in there, and it's going to have different bonds in there, it's going to have beta linkages in there, it takes different enzymes to break that down. So if you look at some animals, say your dog, what's going to happen when they eat something high in cellulose like grass? What happens to the grass? So if your dog eats long grass, out the other end comes long grass. Yeah. So guess what would happen if you ate a piece of long grass? Out the other end would come a long piece of grass in there. So you and your dog do not have the enzymes to digest cellulose. So you can go eat all the grass you would desire, and you're not really going to be able to digest it in there. If you're a cow, you have different bacteria that help to digest that in there. So that's why some animals can do that and others can't in there. So the fiber like that is insoluble, meaning it won't dissolve into things. Another type of structural polysaccharide is chitin. This is in arthropod skeletons, a lot of fungi, also in shellfish in there. So there are some cultures that will eat like shrimp heads. They deep fry them. I tried it. It actually wasn't that bad. It's a little weird and pokey, but it tasted like anything else deep fried. If you put enough fat on it, it's not too bad. It's very crunchy. I don't know that I could go there with others in the arthropod world to crunch them, but that's what makes bugs crunchy like that. So if you step on a big beetle, you notice that it crunches underneath your foot. That's the chitin that allows that to happen. So this one just shows where you've got a slight difference in here in your alpha glucose versus beta glucose in there. Starch, how the bonds are all linear in there, with cellulose, how they kind of alter like that. So can digest it, can't digest it. So your lipids are going to all be hydrophobic, meaning they are not going to particularly enjoy water in there. This is going to include waxes, certain pigments, fats, phospholipids, and sterols in there. So your fats are constructed of glycerol and fatty acids in there. Your fatty acid is going to be a long carbon chain in there. Usually there's 16 to 18 carbons in living systems in there. And at the end of it you have a carboxyl group. So they're relatively nonpolar in there. Everything is symmetrical around it. So when you put fat with water, it doesn't want to mix. The reason for that is the water has that slightly positive end and the partially positive and partially negative. Fat doesn't have that. It's all equal all around it. So they don't like to mix because of that in there. The glycerol is an alcohol that it attaches to with 3 carbons. So what you end up with is this 3-carbon sugar, glycerol, that's going to have a fatty acid attached to each of them. And what that ends up making is a triglyceride. Oh, that was lame. Look at that when it moves, it gives me motion sickness to see the screen move. So the triglycerols or triglycerides, that's what you're going to have most of the fat be in animals in there, is going to be the form of a triglyceride. So most of what you eat is going to be a triglyceride. Everything you store is going to be a triglyceride in there. So when a fat is saturated, it means that we don't have any double bonds between the carbons. They're all attached to as many hydrogens as possible in there. So if we look at our bonds along here, so you got your carboxyl on the end. You've got hydrogens on all of those. It's saturated. However, if you have it unsaturated, it means you've got one or more of these that instead of bonding with another hydrogen, it bonds with a neighboring carbon to get its 4 bonds in there. So you end up with a double bond in there. So monounsaturated means there's 1 double bond. Polyunsaturated means there's more than 1 double bond. So in what you eat in your diet, what they figure we need to have is about 1/3 of your fats be saturated, 1/3 be monounsaturated, 1/3 be polyunsaturated. Again, those saturated fats tend to get a bad rap, and it's not that they're a bad thing, it's that they taste better. So your saturated fats in your diet are usually animal fats in there, things like butter and lard. So if you've ever had like tortillas made with real lard in there, that is, it just kind of ruins all the tortillas in the store. They are really good that way. But it's just the fact that it's good, we have a tendency to overdo it in there. Nobody wants to get up and put some corn oil on their toast in the morning. It's just, you know, it's the butter, yeah, we like that on there loaded up. The corn oil, not so much in there. So when fats are hydrogenated, it's an unsaturated fat that's synthetically converted to a saturated fat. And this is used in the food industry because fats will have a tendency to go rancid, and they will also have a tendency to separate out. When they do this artificially in there, it keeps them from separating in there. So if you look at like natural peanut butter that is just the peanuts that have been ground up, if it sits for a while, it separates. Not really a big deal, you mix it up and it tastes fine. If you look at the other peanut butter like the Skippy or the Jiffy and those kinds of things that don't separate, they have trans fats mixed in there, your hydrogenated oils, to keep them from separating out. So the trans fats in there, part of the process with that is you make things, instead of being sits where the legs on the carbon are next to each other, with a trans fat, now you have them opposite each other. And it takes different enzymes to break that down. So just like you go in cellulose in there, most people, once you see the whole grass incident happen with your dog, or if you try it yourself, corn is another thing. Everyone's probably seen that. There's no arguing that did not break down. Takes different enzymes to do it. Same thing with this. It's going to take different enzymes to do it, even though it looks like a minor difference in there. One, this is what your body is meant to deal with more of. It's not meant to deal with as much of this. So they're also going to have chemical properties that are different. When you artificially hydrogenate things, you get almost all this, where in nature it's more of a mix. And it will change the properties of the cell walls and other things that are made up of this. So that's why now things, for a long time they promoted it as, the margarine's better for you and everything else like that. Now they've realized these actually are worse for you. They're more cardiovascular risk in there. So now you're starting to see things advertised on the package that says 0 trans fats in it. And it's just the change in awareness now that we're like, oh, that really wasn't better for us. So if you have to choose between the butter and margarine, yes, butter is more saturated, but your body knows what to do with it. This floats around too long in there. So the phospholipids, these are going to have 2 fatty acids attached to a glycerol, and then on the third one, you're going to have a phosphate head in here. So this is showing glycerol here in there and a fatty acid attached to it. So you got phosphate, fatty acid, fatty acid. However, they're not in a line like that. The way it's usually drawn is to look more like this, where you're going to have the phosphate head on one end, the fatty acid tails on the other, so that it makes 2 different ends. You've got an end that can actually be okay with water. This end will like water, this end won't. And that's what's going to make a phospholipid really unique, is that ability to get along with water in here. So we use that in cell membranes because you want to be able to have your cell membranes have water inside your cell and water outside your cell, and not be trying to constantly push it away. So the sterols, these are going to be lipids with the carbon skeleton again that has 4 fused rings in it. And they can have different chemical groups attached to the rings. The most common one that you're going to find in animal cells is cholesterol. It's the precursor to making your steroid hormones, and it can contribute to atherosclerosis with the trans and saturated fats in there. So that's another one of those things that you get measured on a blood chemistry panel. If they do a lipid panel, they're going to look at your levels of cholesterol. Plants will have other sterols in them, and the plant sterols, plants don't have cholesterol in them at all, so the other sterols in the plants are used for other things in there. So this just picture shows how with the saturated fats, versus the unsaturated fats. With the saturated fats, they're solid at room temperature. What allows them to be solid like that is they make a nice linear molecule in here that stacks up nice and neatly. So it's kind of like if you look at a ream of paper and they're all flat, brand new sheets of paper, they'll stack up nice and neatly, be relatively stable in a pile. However, if you put a bend in all of those sheets of paper, and not taking the whole pile and bending it together, but each individual paper gets a bend in it, when you go to stack it up, it doesn't stack up quite so neatly. So that means it's going to be less stable, which means it's going to have to be in a liquid state at room temperature in there. So monounsaturated only has one of those. Olive oil is going to be high in monounsaturated fats. It's going to be liquid at room temperature. If you put it in the refrigerator for a few days, it will actually get turned solid. However, things like soybean oil, corn oil, canola oil are polyunsaturated in there. They're even less stable. They're still going to be liquid in the refrigerator. And then this one is showing where you've got the phosphate head, the hydrophilic tails in there for your phospholipid. So next are the proteins, and they're over 50% of the dry mass of most cells. You'll notice what it does not say is protein is an energy source in there. If your body has to try and use protein for energy, it's not giving it the desired fuel in there, and it's not going to work as well. You also get byproducts in there like ketones that will tend to make you smell bad in there. So proteins are made for structures, not as an energy source in there. Yep. So the more muscle you are building, the more protein you would need in your diet. If you're not building a whole lot of muscle, what you need is just enough to repair and maintain structures in there. So that's where different protein needs come in. So things that we use proteins for: enzymatic proteins, structures, storage, transport, hormonal receptors, contractile, motor, and defensive proteins in here. So some examples to make enzymes in there. Defensive proteins, these are going to be things like making your antibodies in there. Structures in there, so the ovalbumin in there, casein and milk in there. Transport proteins to allow things to cross cell membranes. They can be part of some hormones in there, so insulin is an example in there. They can be used as receptor proteins, so when you're going to have a cell send a message to another cell, it has to send something to communicate in there. And what it's going to communicate with is a lot of times a protein in there. Contractile and motor proteins, so your muscles in there. And then structural proteins, so things like collagen in there that fit into other categories. So the polymers in there are all going to be constructed of 20 different amino acids in there. So there's 20 different amino acids that you find in animal cells in there. So a protein is going to be one or more polypeptides. So the peptide or the amino acid is the individual building block in there. So amino acids have a carbon in the center, and they're going to have an amine group on one side. They're going to have a hydrogen on one side, the carboxyl or the acid on one side. So this is why they're called amino acids. And then you've got R, that's your variable group. So in all 20 of them, this part stays the same. The only thing that's different is what is it R in there. So they can get grouped into different things in here with types of amino acids. So you have your nonpolar ones in here, the polar ones, electrically charged that become acidic or basic in there. So lots of different groupings of them in there. The groupings that you would want to know are which ones are the essential, which ones are non-essential, and which ones are branch chain amino acids in here. The last three groupings are the ones that are most significant for what you would use in biology. So the essential ones are ones that you have to consume through your diet. Your body can't make them out of other ones. The non-essential ones, your body can make those using other ones if it needs to in there. And the branch chain amino acids are ones that are metabolized in the muscle rather than the liver in there. So those are more important. A lot of times athletes will look for those, bodybuilders. You'll look and you'll see in their supplements that talk about having more branch chain amino acids in it in there because it's trying to replenish what's in the muscle and help to build muscle. So that's where the significance of those comes in. So your amino acid polymers get linked by a peptide bond in there. So it's a covalent bond formed when the carboxyl group next to an amine group joins together. So what happens is when this has another neighboring one, in there, your bond forms because of these in there. And what's going to happen is you're going to pull off water in there. When you form the bond in there, you dehydration synthesis. So you'll pull off like that, have them hooked together, and that's what forms your bond. And so being that they will line up like that where you have amine end, carboxyl end, amine end, carboxyl end, you can just keep growing the chain in there. So your protein structure and function. Proteins function based on their 3D shape in there. So we have different levels of the protein structure. Your primary structure is just the basic sequence in there of different amino acids in there. So what order are the amino acids in if you break them up in a long chain? But then they have a secondary structure in there where they're either going to form these beta pleated sheets or they're going to make these alpha helices in there. So they will form one of those two shapes. And then we have it go into a tertiary structure where it's going to fold up even more with that. And the quaternary structure is a lot of times you're going to have more than one polypeptide come together as subunits of a bigger protein in there. So some of them are going to be globular proteins where they end up being roughly spherical in shape. Some will end up being more fibrous proteins that will be shaped more like long fibers in there. So when you make the tertiary structure in there, a lot of times what's going to help hold things together is hydrophobic interactions in there, little clusters in the core that are out of contact with water. Another thing is the disulfide bridges. So where you have cysteine, cysteine has sulfur in that R group in there, and the sulfurs will make these sulfhydryl groups in there and end up having these sulfur-sulfur bonds together. Those sulfur-sulfur bonds are really strong, so that's going to make up a lot of structures. Some of your collagen is going to have that. Your harder proteins are going to have more of those disulfide bridges in there. So things like hair, fingernails are going to be high in the sulfur content in there. So this just goes through and illustrates some of the different structures in here with your primary structure here. And this is showing for blood cells in here. So with that primary structure in there, that's just the order of the amino acids that it creates this particular shape. If you change just one of the amino acids in there, it can change the entire shape of the protein. And that's what happens with sickle cell anemia. So normally you're going to have your hemoglobin have these alpha and beta subunits that are in normal shape in here, and it makes the red blood cell be this shape. However, with sickle cell anemia, one of these is off, and so it changes the shape in there, and what you end up with is these sickle cell or sickle-shaped red blood cells in there that are not going to fit through blood vessels the same in there, which will cause the problems for the person with the sickle cell anemia. So this order in here, if this order is different, it ends up changing it all the way through. Nope. So when a protein denatures, this is when it's going to lose its shape and becomes biologically inactive in there. Sometimes this can be reversed in the lab under controlled conditions, but most of the time it can't. So an example would be if you fry an egg. So when you put the egg in the pan, you've got the yellow part that's liquid, and you've got the white or the clear part that is mucousy liquid in there. When you heat it up, you're going to denature it, and it turns white. You can't uncook the egg. So if you walk away and you're making your eggs and it gets a little too cooked, you can't undo that. It's permanent in there. So that's an example of denaturing things. In the body, a lot of proteins will actually denature at about 105 degrees Fahrenheit in there. So when they look at what temperature does it become really critical in there, if you're watching somebody with a fever, at 105 is where you can potentially have serious damage occur in there because of proteins denaturing. So chaperonins, these are chaperone proteins that assist in proper folding. So you have these sequences like this that are long sequences that always fold up into the same shape. If it's the same order, it will always make the same shape every single time. And what allows that to happen in there and helps make sure that happens is the chaperonin proteins that are going to help guide it in there. So how do we know what the shapes of these things are? X-ray crystallography has been used to determine the 3D structure in there. So they used an X-ray and put it through a crystal of it and basically took an X-ray pattern of it to be able to see the things in there. Now they use computer models to do things in there. So nuclear magnetic resonance spectroscopy was another method that didn't require them to be formed into crystals. We know the sequence of enough things now that we can actually predict what shape it's going to be just with a computer model. So last group, your nucleic acids. So here we have the genes, which are the units of inheritance. They're made up of DNA, and it's going to determine the primary sequence of the polypeptide. So that DNA is the instructions for making everything in your body, because the order of the DNA tells you what order to put the amino acids in the structure that you're going to make in there. So DNA is a deoxyribonucleic acid, RNA is ribonucleic acid. You would almost always see them abbreviated as DNA and RNA at this point. I think they're probably even in the dictionary that way without having to be all the way spelled out. So what a chromosome is a long strand of DNA that's going to carry 100 or more genes, except if you're a Y chromosome. And it's the organized form of genetic material that you have when the cell divides. So it's a long, organized, coiled up piece of DNA in there, is what a chromosome is. Y chromosomes don't have over 100. Right now we think the Y chromosome has 78 genes. That's a man. And it's the only one that's just a little tiny one in there. The Y chromosome's kind of a different one. Actually, what determines whether or not you produce male parts is only one gene on the Y chromosome. So on the chromosome like that, it's what's going to determine, the one gene will determine, do you make a penis or do you make a vagina? Everything else about sexuality is outside of there. We don't really know where that comes from in there. It's a lot more complicated than what just saying which parts you get.
So what happens when you have a hermaphrodite?
That's actually two different zygotes that fuse together in there. So your mRNA is your messenger RNA, and that's going to be when you do protein synthesis, you don't actually use your DNA to do that. The DNA has the instructions, but that's kind of your master copy. It's like saying, I'm going to make a photocopy of this to give you on the instructions in there. That's what the mRNA is it's a RNA copy of it that is actually used by the ribosomes, your organelle that does the protein synthesis in there. So the kind of order of things is you start with DNA, you have to make an RNA from it, and then you make the polypeptide or protein from it. So a polynucleotide is just a polymer of nucleic acid macromolecules. A couple of terms you'll see in there, nucleotide and nucleoside in there. So a nucleotide has 3 parts to it in there. It's got the nitrogenous base, a 5-carbon sugar, and a phosphate. A nucleoside is just the pentose or 5-carbon sugar and the nitrogenous base, but no phosphate on there. And the only way that I can keep track of it is I think, okay, you've got S and then you've got T in the alphabet. T is afterwards. The smaller one, the bigger one in there. Okay, yeah, that would work too. I mean, otherwise to look at that and be able to get it without making some little trick to remember it, it's one of those things where it probably just isn't going to happen because they're kind of weird words like that. So your families of nitrogenous bases, they get grouped into being a pyrimidine or a purine. So the pyrimidines have 6-membered rings of carbon and nitrogen, and it's going to include C, T, and U, which stand for cytosine, thymine, and uracil, which are what you see up here. So cytosine, thymine, and uracil. So you'll notice the pyrimidines have just 1 ring. The purines are bigger, and they're going to have 2 rings in them. So it's going to be adenine and guanine. And then they're going to be attached to the sugars, either deoxyribose or ribose. So what's going to happen is you're going to have purine will bond with a pyrimidine in the structure in there. So when you look going across the DNA, you get a total of 3 rings in there. So the atoms in the nitrogenous bases are numbered so that the sugar atoms will have prime after the number to distinguish them. So you'll see it being referred to as the 5' carbon in there. And so it's just a way of marking where do we start the circle at when we're counting the carbons in there. It's just something that they've arbitrarily chosen in there. So when we glue these together, we use a phosphodiester linkage in there to join the adjacent nucleotides. And so you're going to have a phosphate group in there linking the 2 sugars. So your backbone, which is like the sides of the ladder, ends up being a repeating pattern of sugar and phosphates. So at one end you're going to have a 5' end in there where you're going to have a phosphate attached to the 5' carbon. At the other end of it you have the 3' end, which has a hydroxyl attached to the 3' carbon in there. So that's how we determine which end when the DNA is which, a 5' end and a 3' end in there. So the reason that is significant is because DNA is going to build in the direction from 5' to 3'. And that's how it's going to read, is that 5' to 3' direction in there. So the gene's meaning is actually encoded in that specific sequence of the 4 DNA bases in there. So whatever order those DNA bases are determines what amino acids will be in the protein. So DNA is going to be as a double helix in human cells. You can have this vary in viruses in there. It's a little bit different. So your RNA molecules are going to be the single polynucleotide chain in there. So RNA is usually single-stranded in humans and animals in there. So when it's the double helix, it's like a ladder that's been twisted. And we say that the arrangement is anti-parallel in there, so it's going to go 5' to 3' on one end, and then 3' to 5' on the other end. So in this picture, you can see 5' to 3' on one side, 5' to 3' over here. So they run kind of like this. So when our bases are going to pair, in DNA, A and T pair together, G and C pair together. So the two strands are complementary. And so this shows here where you've got A and T, G and C. The way they are indicating this is you've got round ends and pointy ends. Rounds have to go with rounds, pointies have to go with pointies. So in RNA, you're going to have A pair with U for uracil instead. Uracil is similar in shape to the thymine, but it's only found in RNA. And then the G and C are still the same. So because of this, if you split that in half, there's only one possible thing that can match up with the other side. You can split it in half and use that as a means to duplicate it in there. So this is just kind of a big picture of what happens. You start out with the DNA that's got your instructions when you want to make a protein. You're going to make an mRNA copy of it. That will go out into the cytoplasm. The ribosomes will hop on there and read it, make the amino acids, or they will take the amino acids and assemble them into the polypeptide that you're making, which we're going to go into more detail of that later. But that's just if you want to look at it and you go, so where is this DNA stuff going? Why are we doing this? That's what's coming down the road with it. So DNA and evolution, molecular genealogy. We expect that two species that appear to be closely related on the fossil and anatomical evidence are going to share a greater amount of their DNA and protein sequences. And for the most part, we've been pretty good at estimating that, but some things are getting reclassified into different relationships with looking at their DNA sequences. Not so much in the big stuff where you're looking at the animal world where we can see it with our eyes and we've seen visual differences, but a lot in the microscopic world with like bacteria. Because pretty much to our eye, even underneath the microscope, they all look the same. So we've had to look at other ways to be able to try and identify how they're similar in there. Siblings have greater similarities in DNA than unrelated individuals of the same species too. So let's go ahead and take a break for a few minutes.